Hello students, my name is Shashank Shekhar. So today is the third lecture of the chapter Biomolecule. So far we have studied what is carbohydrate, what is monosaccharides, what is glucose. We have started glucose, we have not studied a whole about glucose. We have seen the chemical reactions, we have seen some of the properties of glucose. So we are done with this thing. Now today we will move more appropriately into this glucose. We'll know in depth what this glucose looks like when it is present in solution. What is the physical structure of this glucose and why the reaction that it gives are in this. So let us start our today's class. So if I talk about the syllabus, so right now we are here, that is in glucose. So if I talk about the topics, so we will see what is the cyclic structure of glucose. Okay, after that we will see how is the cyclic structure formed. Then we will see its say resemblance with pyranose. After that we will see two forms of cyclic structure and how they are formed. At last we will see glyceraldehyde. So glyceraldehyde is also a carbohydrate but with lesser number of carbons. Clear? Now let us start our today's class. So first of all, why is this cyclic structure being proposed? What is the reason behind it? So the reason behind it was that some of the chemical properties were not in accordance with the structure that was proposed. Means the structure was not able to explain all the chemical properties that the glucose show. So for that, the cyclic structure was introduced. So what are those chemical reactions? The first one is that glucose does not give Skiff's test. Glucose does not give Skiff's test. What is Skiff's test? So in Skiff's test, if we treat our aldehyde with Skiff's reagent, then it gives magenta color. Now if I talk about the mechanism of it, it is nothing but a nucleophilic addition reaction. Now you would say, sir, we have seen earlier that our glucose was giving nucleophilic substitution reaction, but now what is the problem? See, the problem lies in that it is a weak nucleophile and it will only attack if there is free aldehyde. So free aldehyde and attached aldehyde are two different things. Attached aldehyde can give reaction with stronger nucleophiles but not with the weak nucleophile. So why it is a weak nucleophile is a Skiff's reagent. So it is basically NH. So it is basically N here, SO3H and H, and this N is being attached to benzene ring, and we have more R here in this position. So we'll not talk about that. So if you see here, its lone pair are delocalized. It is delocalized in the ring. What will be the result of this? That this lone pair will not be available for nucleophilic attack. So due to this reason, due to this conjugation, I would say the nucleophilicity of this nitrogen has been reduced. Okay, clear? Therefore, if you can say favorable conditions are not present in the say structure of the aldehyde, then it will not be attacked by the Skiff's reagent and no magenta color will be given. So due to this reason, we have say raised the question that why it is not giving Skiff's test. So it must be lacking a free aldehyde group. Next is, if we treat our pentaacetate of glucose. So if you remember what is pentaacetate of glucose, so in pentaacetate of glucose was formed by the reaction of our glucose with acetic anhydride. To give our pentaacetate glucose. So in that structure we have, if you remember, we have CHO here and each of this says OH has been replaced by OCH3. OCH3. Similarly, all of the O were changed to this. Now, what was seen that if you treat your pentaacetate with hydroxylamine, then it would not give this reaction. Okay, the pentaacetate does not react with hydroxylamine, indicating the absence of free CHO group. So, absence of this reaction, that is your reaction of glucose with means pentaacetate of glucose reaction with hydroxylamine, indicates that we are lacking CHO structure in your aldehyde. Next, what was the evidence? The next evidence was that our glucose is existed in two forms. First is the alpha form and second is the beta form. Now, if I look at the structure of both of this alpha and beta form, then it has no signs of a free CHO group. Means you will see in the later on slide that the CHO group is being replaced by our hemiacetal group. So all of these behaviors, like the Skiff test, the reaction of the absence of reaction of say pentaacetate of glucose to give say our final product, then the existence of our glucose in two forms, the alpha form and the beta form, namely, which have a different melting point. Like our alpha form has a melting point of 419 Kelvin, and your beta form has a melting point of 423 Kelvin. And they both were obtained by the crystallization of hot and saturated aqueous solution. Now, all these evidences suggested that our glucose is not existing in the form of simple chain length, or you can say open chain. So for that, we have proposed a new cyclic structure, and it was termed as cyclic hemiacetal structure. And what was the proposition? How was the cyclic group formed? It was proposed that the free OH group, so if you see here at the structure, so what must have happened, that our free OH group at the fifth position, so here we have free OH group, so it must have attacked this carbonyl center and formed a ring. Now, I would not say that our glucose would have always existed in say this form or this form and this form. There is a kind of equilibrium in all these forms, and each of these has some attribution to our actual structure. Okay, clear? So attack can happen in two ways. So if I see the structure, you can imagine then it can attack from the upside and from the bottom side. Okay, that would result in two type of structure, namely alpha D glucose and beta D glucose. And this is called hemiacetal. Why is it called hemiacetal? So if you remember the aldehyde, ketone and carboxylic acid chapter, then we have seen that if I attack a carbonyl center, more appropriately I would say aldehyde center with a hydroxyl group, with H replaced by R. Okay, H replaced by R. Then this alkoxide will attack the carbonyl center and will form OH and OR. So this group is called as hemiacetal. Clear? Did you get that? So you can see here that our OH group at the fifth position is attacking this carbonyl center, and this is forming a hemiacetal. We have OH here and O here. Similarly, we have OH here and O here. And the R is this thing. Although it is intramolecularly linked, but it is R. Now, both of this structure explains why we are not getting certain reagent, or why we are not getting certain reaction, because the equilibrium is mostly shifted either to alpha or either to beta. This structure is not that much effective to explain the attributes of our glucose. Now, also it explains the existence of two types of structure of glucose, this alpha D glucose and this beta D glucose. Now, what is the difference between alpha D and beta D glucose? So if you look at if I talk about the spatial arrangement, if you look at the first carbon, you can see that the OH group are present at different positions. in beta D glucose, our OH group is present at the left side, and our alpha D glucose, our OH group is present at the right side. So you will get a more clear picture when you will see the actual structure, the cyclic structure of it. Now one more point which I mentioned earlier also, that you should remember this, that this all three structure are in equilibrium with each other. So some of the, say, our reactions that were given earlier, like our nucleophilic addition and substitution to this carbonyl center, were the results of this equilibrium that existed between all of these three structures. Okay, clear? So in this way our cyclic structure is formed. Now we have a resemblance of the cyclic structure with pyrene. So this is called as pyrene. So for that reason it is called pyranose. So what is pyrene? It is basically a heterocyclic compound with oxygen as one of the member of the chain, cyclic chain, and we have 4 sp2 hybridized carbon and 1 sp3 hybridized carbon. So if you see and try to, say, compare both of the structure, you will see that they are similar. We have oxygen here and we have the alkyl chain surrounded it, making a cyclic chain here. So I was talking about alpha and beta D glucose. So there in the Fischer projection it was not that much clear, means the spatial arrangement was that much not that much clear, but you would see here that we have a clear picture of why this spatial arrangement different. So if we place this say our cyclic structure on a plane and keep it in such a way that our CH2OH group is present on top, then you will see that the OH group in case of beta D glucose is present on the upside. Okay, it is present on the upside, while in the alpha D glucose it is present on the downside. So if I ask you what in what way is the attack happening, so in this case the OH group has must have been attacked from the upside. So it has been this carbonyl center has been attacked from the upside. Due to this reason this OH group went down or has gone down, and we have it has resulted in alpha form. Whereas in the case of your beta, this OH have attacked from the downside and it has formed this beta. So these are the two structures of, say, our cyclic structure of the glucose, and it was, say, represented by Haworth projection. This is called as Haworth projection. Now if I ask you that which of these two structure, out of these two structure, which is the stable one, what would be the answer? So for that you have to look at the steric hindrance part. So let me clear all this. Okay, so here we have a CH2OH group, which is a bulky alkyl group that is present on the upside. Here also we have the same group. Now if I compare both of these, you will find that we have an OH group here, which is in comparison to H, which is more bulky. So if it is present on the downside, so two reasons are there for the say presence of OH group on the downside. First of all, I would not say steric hindrance, more appropriately I would say that both of this OH group now can do the hydrogen bonding. Both of this OH group can do the hydrogen bonding. Due to this reason, both of this OH group are present on the same side, and it is found that alpha is the more stable one. So this is the structure of our cyclic structure of the glucose, that is in resemblance with the pyranose. So what are the points that you should remember? First of all, you must remember the numbering of this. It is first, second, third, fourth, fifth, sixth. So at the first position, so the first position is deciding that which form of the glucose are we looking at. So if the OH group is present in the downside in the first position, then we call it as alpha D glucopyranose, and if it is present on the upside, we call it as beta D glucopyranose. Now one more thing. So we have seen the classification of our sugar in two types, the reducing and the non-reducing one, and we said that the presence of OH group, free OH group, would decide our, say, reactivity, or you can say reactivity towards our Benedict solution or our Tolvin stage or our Fehling stage. So the free OH group are deciding at the first position. So you can see here we have this as the first position and we have OH group present and it is free. How it cannot be free? If I ask you how it cannot be free, suppose it was attached to this CH3 group. Then in that case, it would have not given our failing solution test or Tolvin's test. But since we have a free OH group here at the first position, so it will give our failing test or the Tolvin's test or the Benedict's test. So in exam, if you are given a couple of structures and you are asked that say which one of these are reducing and which one of these are non-reducing, so you will simply look at the first carbon that is being next to the oxygen and we'll see that whether a free OH group is present there or not. If it is present there, then it will give the reducing test, and if it is not present there, it will not give the reducing test or will not convert or not reduce our failings, Tolvin's or Benedict solution. Clear? Now both of these are called as anomers of each other. So this is just a stereochemical aspect of our glucose. It is just a stereochemical aspect of our glucose in which the position means the position of the OH group has been changed. So this is called as anomeric carbon and both of these are called as anomer. Now if it is changed at first position, it is called as anomer. If it is changed at any position, that is called as epimer. So if I am saying this both are epimer and anomer, so it is correct to say that. But when we are saying it is epimer, we have to define that at which carbon the position is changed. So we can say that at C1 position, due to the change in configuration at C1 position, this both are epimer. So it can be termed as alpha D glucopyranose and it can be termed as beta D glucopyranose. Okay, got it? Now let us see some simpler carbohydrates. So this is called as glyceraldehyde. Now if I ask you under what classification does it falls? Under what classification does it form? On the basis of both on the basis of carbon number of carbon atom and on the basis of carbonyl center. So let us first look at this. Okay. So how many carbon it has? 1, 2, 3 carbons. So 3 carbons means triose. This is also 1, 2 and 3 carbon. So it is also triose. Next thing, what is the position of the carbonyl center? If it is present at 1, it will be aldose. If it is present at 2, it will be ketose. So since it is present at 1, so it will be termed as aldose. Similarly, it will also be aldose. So this both are aldotriose. Aldotriose. So glucose was aldohexose and this one is termed as aldotriose. So this is the smallest or the simplest member of our carbohydrate family. Now let us try to remember, or I will give you a trick to remember all of the structure of our, say this aldehyde, carbohydrate. So for that you should remember this chart. You should remember this chart. Now how you will remember it? For that we have a trick. What is the trick? First of all, try to remember this thing that we call it as get Raxel. One more thing I would ask you. Why are we saying D? Why are we saying D here? Why are we saying D here? We have seen that we have said it alpha D glucose, beta D glucose. Then what is the L? Okay, so for that you should focus on the carbon that is present at this position for all of this, at this position. Now you will see in all of this that the OH group is present on the right side. OH group is present on the right side. So if your OH group at this carbon that is just next to CH2OH is present on the right side, then we will call it as D form. We'll call it as D form. And if it is present on the left side, say it is present on the left side, so if I draw a similar one, O, H, C, OH, I am putting this OH on the left side, and CH2OH. So in that case, we will call it as L-glyceraldehyde. Similarly, if we are asked to write the L form of this, how we will write? We have C, we have OH. We will place this. So I will repeat it again. You will have to change the position of this carbon only. Means the OH position of this OH group only. So we'll write it in this way. So you can see I have left this thing as it is, and I have just changed the position of hydroxyl group present at this carbon. So this will be termed as L-threose. Similarly, for this, if I would say, then it would be CCC, OH on this side, H on this side, and CHO. You can again see that I have placed OH on this side only, and the only thing I have changed is this position of this hydroxyl group. I have not disturbed the position of any other hydroxyl group. Similarly, you can change this position and get your L glucose. Okay, you can change this position and get your L glucose. Now, if I ask you under what classification do they fall? So all of these are aldose. Why? Because the carbonyl center is at the first position. Carbonyl center is at the first position in each of them. Okay. Now let us try to divide it on the basis of number of carbon atoms. So we have seen this thing. This is glyceraldehyde, it is triose. Or what are all these things? This is 1, 2, 3, 4 carbon. So it is tetrose. Again, we have 5 carbons here. So it will be pentose. 6 carbons, so hexose. So this will be aldotriose, aldotetrose, aldopentose, and aldohexose. Now initially, I have told you a trick to remember the name of all of this, and I would say that it is important to get a grasp on the names. Okay, so it is important because they are common names and there is no such convention, IPC convention, to remember the name of this compound. So the trick for that is get Raxal. GET, get RAXAL. How you will follow it? So first of all, take the letter G, which forms glyceraldehyde, and it has 3 carbons and only one say stereochemical center or the optical center, which has OH on the right side. Now if I move down the flow chart, then we will follow this get Raxal. We have E and T. So E stands for erythriose and T stands for threose. Okay, D stands for erythriose and E stands for erythriose and T stands for threose. Now if I ask you what is the change in this and this, what is the distinction in this and this structure, so you will say that on the second carbon we have a change of the spatial arrangement of the hydroxyl group. So in this case our OH has been kept on the right side, and in this case our OH has been placed on the left side. So what we have done, we have just introduced one more carbon. We do not have to do anything with the carbon that is present on the bottom side. Only the carbon that has been introduced will have change in spatial arrangement. So if I see at the left side, the OH group on the right side. If I see on the right side, the OH group on the left side. So as we proceed down and say we put 2 subdivisions of that, then the subdivision on the left side will have the OH group on the right side, and the subdivision on the right side will have the OH group on the left side. And whose OH group? The carbon that we have introduced or the carbon that we have increased. Okay. Now next, if we have erythriose, if you move down to erythriose, we have ribose and arabinose that come from R and A of Raxel. So ribose and arabinose. Again, we do not have to do anything with this carbon, the remaining two carbon. So we will only change the position of the OH group that of that carbon which is present on the or which is being increased. Here you can see that we have introduced this carbon. So therefore the group on the left side has been placed with the OH on the right side, and the group on the right side has been placed with OH on the left side. So we are done with GET, we are done with RA. Then XL. So with threose, again we will divide threose into two, xylose and lixose. So in this case also we will not do anything with the carbon center that is present in the bottom. Only we will change the position of the OH group that is attached to that carbon which has been newly introduced. So this is the newly introduced carbon. So if I see on the left side, the OH group was on the right side, and again this is on the right side, so OH group will be on the left side. So we have done this far with the help of Gatraxel, so xylose and lixose. Now how to remember this? All allose, ultrose, glucose, mannose, glucose, idose, galactose, talose. So for that we have this trick which is said as all ultrious. Let me write it. All ultrious gladly make gum in a giant tank. So let us follow this. So we have all, first of all. So from all comes the allose. All comes the allose. Again we will subdivide this ribose. We will not say change any of the position that is related to the carbon on the bottom side. We will only change the newly introduced carbon position. So again the left carbon will get the hydroxyl group on the right. So we have allose. Then all ultrious. So from ultrious comes the ultrose. Now since this group is on the right side, it will be termed as means the hydroxyl group will be present to the left side. All ultrious gladly. So from gladly comes the glucose. Gladly comes the glucose. So in this you can see we have this arrangement. It is being retained here. So this arrangement has been retained here. Only the OH group of this carbon has been changed. So on the left side we have on the right side OH group on the right side, and again this mannose comes from made. So all ultrious gladly make. So makes introduce the mannose, and again this has been retained. This group has been retained, and this position, this carbon position has been changed. Now if I ask you why are we studying this? Why are we interested in glucose, mannose and all of this complicated structure? So you will see in the further reactions that they are showing some reactions. I mean some of the reactions will show the same product. Means if you react your glucose and mannose, if you do this reaction, a specific reaction, then both of this will give the same reaction, same product. So what is the benefit of this? So see students, some of the questions are framed in this way that say this is the compound, it reacts with this to give this and this. So for that case, if you are not aware about the say structural attribute of a given compound, then it will then you will face difficulty to get your answer, and you will lose one of the say hint that is given in the question to get the proper compound. So for that reason I would suggest you that you try to get a grasp of all of the structure, because that would be helpful to you throughout the chapter. And this whole of this biomolecule is all about structures. Starting from the cyclic structure, we have different spatial arrangement of OH group. then we have the Howard projection, then we are moving to this different type of, say, carbohydrates. So we are done with the mannose. Then we have the xylose. So xylose again, so gladly make gum. From gum comes our glucose in a giant tank. From in comes our idose. In gives our idose. Again, the retention of the carbon center that are present on the bottom side and change in the OH group which is being attached to the carbon that has been newly introduced. Similarly for this, so all ultros gladly make gum in a giant tank. So this is the overall table that you should try to remember. So we are done with this, say, structural part. Now when we will move to, say, our next thing, that is ketose, then we will track this again. Okay, so in ketose, if you remember, the carbonyl group was present on the second carbon atom, on the second carbon atom. Now let me frame a question for you. Say if I say you that which of these compound will give the same product upon reduction with HI, which group of compound? So if you see here, if you reduce all of these with HI, you will get an hexane. An hexane. Similarly, if you reduce all four of this, you will get an pentane. Again, if you reduce these two, you will get an butane. Then at last you will get propane. N means normal chain. We do not have any, say, our substituents present on pentane. It is just a normal chain. Then we have N-hexane, normal hexane. Okay. So this was about the various classifications of our, say, glucose. Now we have seen the cyclic structure. So if I try to summarize our today's class, so what we have seen that some of the chemical reactions are not being justified with the help of open chain structure than we had. Starting from the Skiff's reagent, we do not get any magenta color upon treatment of our glucose with our, say, Skiff's reagent. Then our pentaacetate glucose does not react with, does not give nucleophilic substitution. Okay. Then the presence of two type of, say, our glucose, that is alpha D glucose and beta D glucose. So upon crystallization of glucose, we got two structures. So those two structures were not explained with the help of our open chain structure. So all of this combined give us an idea that our actual structure, open chain structure, is not the only structure that is present in the solution of glucose. So for that, we have introduced a cyclic structure. That cyclic structure was further converted into Howard projection. That is a different thing. So that was the structure. So I would tell you one more thing here. Okay, I will tell you one more thing here. So let us talk about, say, this structure. More about the structure. Now, if I ask you that if you try to draw the chair form of this, then how I will draw it? Okay, I will draw it. So let us see. So we have, first of all, we will try to draw the chair form. So this is the chair form of our, say, cyclohexane. Now let us try to put the substituents on this. So at the first, so I am naming this at the first position. So this is the first position. So we will have an O here. We will have an O here. Now H is present on the upside and OH is present on the downside. Let me draw it in a different way. Sorry. Yeah, I'm placing it here only. So we have this H here, and on the downside we have our OH here. Okay. Then on this position we have our OHH here. So at first position we are naming it as the first position. This is the second position. This is the third one, fourth one, fifth one. Now at the fifth position we have the CH2OH. Therefore it will be named like this, CH2OH, and we have H here. Okay, HOH again. Then we have OH and H here, and H here. Yeah. Now if I ask you, is it the stable form? Is the given structure stable? No. Why? Because we have a CH2OH group and there will be 1,3 diaxial interaction. So this is first position, second and third. So there will be 1,3 diaxial interaction. So for that reason, this will flip. So there will be a ring flip, and we will get this thing. Again, let us draw it. H, OH. So you should always remember that our bulky group should always be on the axial position, not on the equatorial position. So we will flip this ring. So this is the sign of ring flipping. So we will get here HOH, and again OH and H. Now if I place the CH2OH group, this will go like this. CH2OH group. Now since it is present at the axial position, so this is the more stable form of the given glucose, our alpha D glucose. Similarly, you can try to draw the structure of the beta D glucose in a similar fashion. So you will do the flip again and see that which of this is stable. Now why I am drawing this? So we will see the anomeric effect. Then you will find that the anomeric effect is kind of being associated to our cyclic structure, and this is the actual structure in which our cyclohexane exists, and this will be the structure of glucose also. So for that reason, I would suggest you that you should be aware about the cyclohexane form. So I will give you a more clear picture of the cyclohexane in the next lecture. Now we are done with this glucose, and we are only left with the cyclic structure of glucose in the cyclohexane form or the chair form. So with this, I would end my lecture. Thank you.